Okay, so today we're going to talk about how DNA is replicated. So as before, I would like you to take notes on this video and we'll discuss it in class tomorrow. So here we have a DNA structure, and we know that DNA is in the form of a double helix, and the rungs of the DNA make up the bases, and this part here along the outside make up the sugars and phosphates. So when we replicate DNA, we're going to start off with one parent cell, and we wind up with two daughter cells that are genetically the same. So if we look at this DNA this DNA structure that I have drawn over here, and if we apply this to mitosis, we could see that we have one chromosome in this particular cell, and by the end of mitosis we're going to have the same copy of this chromosome in each of the two daughter cells. So these two daughter cells that were created will both have the same exact DNA structure. So how does this happen? Well, the first thing that needs to happen is that the DNA, or this double helix, needs to separate. So they're going to separate. So we're going to get each half of the helix on opposite sides of the cell, and then the way how it unzips is with DNA polymerase, which is a type of enzyme that's going to cut the hydrogen bonds between the bases, and free nucleotides that are in the nucleus will start to bond or start to form bonds with the nucleotides of each part of the helix. So over here we're going to get a base that will have a base that will bind to adenine. So we'll have a thymine, we'll have a cytosine going to, we're going to have an adenine over here. Right, T goes to A goes to T, C goes to G, G goes to C. And we're going to wind up with another double helix. And then over here the same thing will happen. T goes to A goes to T, G goes to C, C goes to G, C, and T. And then now if you notice, we have two DNA structures, two double chromosomes, exactly the same. When the cell divides, each one will go into the new cell, and we have two cells that are genetically identical.